I'm Brianna. I'm a senior here at Beaumont, and while I was at the Harrington Discovery Institute this summer, I helped with the research of novel S nitrosylating agent to improve the preservation of human cornea tissue for transplantation.
Next.
I'll be the battery.
So an overview of my PowerPoint is that I will be talking about cornea transplants, brain death, and also snow. So what is a cornea? A cornea is the eye's outermost layer, and what are reasons that people may lose their sight? They could have a trauma, a degeneration, or also reduced endothelial cell density. So for cornea donations, they help to restore or preserve their sight, or a person can also donate to two people. So here's a picture of what our corneas will look like. We often got 2 pairs of corneas a day. Sometimes we might have got them twice a week, but this is what the packaging looked like. So the need for corneas. There are many benefits to cornea transplant, but there's also still a shortage in the U.S. for donors. So the wait time for a span from 6 to 2 years with an average of 22 months. So what is brain death? Brain death is the irreversible loss of brain function, including the brainstem, and it also occurs when intracranial pressure increases above the blood pressure, and this causes physiological harm. So snow, it is an important regulator of bodily functions. It stands for S-nitrosyldiols, which is they control the important body functions, including oxygen delivery and cell activity. So there was a previously conducted study on brain death from the lab that showed that snow levels are disrupted and are related to markers of injury, organ dysfunction, and reductions in oxygen delivery. So these disorders continue during the storage phase for commonly transplanted organs and tissues, and this also includes for corneas. So a possible solution to increase snow. Using a drug our lab has designed, we found that the snow problems could be reversed to reduce tissue injury. And we also want to now see if this treatment of the donor cornea tissue can increase snow levels, the length of storage, and improve the tissue function in the recipient. So the hypothesis that we had is that we hypothesized that enhancing bioactivity, snow, during a corneal donor storage can maintain human corneal endothelial cell function and prevent the decline in endothelial cell density. So we had 2 aims during this experiment. Aim 1 was the characterization of the endothelial snow proteome in stored human donor corneas, and aim 2 was testing if snow therapy can extend storage duration to at least 21 days. So the 3 methods that we used during our experiments were the non-invasive microscopy, which is a way to measure endothelial cell density, and this was done at the Eversight Reading Center. And also we did an assay storage solution, which was checking for levels of snow in the storage solution that the corneas came in, and this is also a chemical analysis of snow. And the last method that we used was biochemical analysis, and this was using a Western blotting test to quantify the expression of NOS1, 2, and 3, which are the 3 proteins that are found in NO in the cornea tissue samples. So here's a picture of what the Western blot test will look like. We will fill these columns up with the samples from the 8 corneas, and then we will run the test, and we will transfer the gel to a membrane, which here's a picture of the membranes cut, and they're being rinsed in solution so we can later develop them. So here's results. I'm sorry for the bad quality, it's very dark, but this top line, which is the same throughout all of them, that's NOS1, and then the second one underneath is NOS2, and then the third blue line is NOS3, and then the pink line is GAP-DH, which is a control for the proteins, and you can see by the highlighted box that the black bars show that there's a lot of protein in these samples of corneas that we ran the test on. a graph of our preliminary data. So the B corneal endothelial cell density after 14 days, the ECD decreased by 60%, and when there's less ECD, this means that there's less healthy cornea tissue. And then also there's a lot of variety. So the average ECD decreased by above 60% after 14 days. And some corneas maintain high ECD between 2,000 to 3,000 over the course of 14 days, while others have a total cell loss, which means they have no endothelial cell density by day 7 or 14. So here's a graph of corneas A and B. Corneas A were the corneas that we ran the test on, while cornea B were the corneas that we left untouched so that we can compare the results of our test for their proteins. So in NOS3, there is variation, and the average NOS3 expression decreases slightly in the B corneas compared to the A corneas. And then for NOS1 variation, the average NOS1 expression decreases slightly in the B corneas compared to the A corneas. So during my time at HDI, I was presented with many new experiences. I never could have imagined myself working in a lab at the age of 17, and I also had the opportunity to cut corneas and run tests on a day-to-day basis, and I also got to run them like myself after a couple of weeks. I enjoyed working in this lab. It was very relaxed and fun, and my experience this summer allowed me to realize that I enjoy working in the lab, and this may become my future job as I hope to become a biomedical engineer.
My name is Colleen Martin. I'm a junior. I was also a fellow intern at the Harrington Discovery Institute this summer, and my project was S-nitralization, microcirculation, and tissue oxygenation in swine brain dead tissue, and I was mentored by Dr. Lin Zhu. So, organ transplant is a treatment of choice for many end-of-stage organ diseases, and a lot of these transplantable organs come from brain-dead individuals. But the problem is that often these organs fail or deteriorate and become unfit for transplant. So, organ transplant is negatively impacted by inflammation and reduced perfusion, which is the flow of blood through the vessels, and both processes are regulated by the protein S-nitralization. So over the summer, to mimic brain death in swine, we would induce the pig and increase the intracranial pressure over the systolic pressure and monitor overnight. And while monitoring, we would monitor the vitals, including the pulse, which is microcirculation, and tissue oxygenation, which represents the microcirculation, so the smaller level and the bigger level. So the hypothesis throughout the summer was that brain death negatively impacts swine tissue microcirculation and leads to changes in biological activities. So this was done through 3 methods. The first method was testing the difference between the pulse oximeter and Hutchinson Inspectra SpO2 monitor. The Hutchinson Inspectra tests tissue perfusion, which is the smaller areas, so that's that microcirculation, and the pulse oximeter monitors oxygen saturation, so that's the greater area, that macrocirculation. So here are the results from the first method that was performed. After monitoring the pig on the pulse oximeter and the Hutchinson Inspectra SpO2 monitor, you can see that there was a major decrease in brain death at the time in both cases, so that also supports the hypothesis. The second method was testing snow levels in the different samples, so the BD samples, the brain dead samples, and the CT samples, the control type, the before brain dead. And so what is snow exactly? I know Brianna touched a little bit on this, but snow is a nitrogen monoxide group attached to the thiol side of a cysteine chain, and it controls cellular functions like apoptosis, cellular metabolism, and membrane trafficking. So my data shows that there was a decreased amount of snow levels in the BD versus the CT. And the depicted here is the machine that a technician at the lab runs the levels.
And the BD and the CT
BD is brain dead and CT is control type 2 before brain dead. So the tissues and samples that were before brain dead are represented by this graph, and tissues that were tested that were taken after brain dead are represented by this bar in that graph. And so in the third method that was performed was a Western blot. So a Western blot is a protein analysis test, and it includes preparing the sample, it includes performing a protein assay, running a gel electrophoresis, blocking, washing, incubating the primary and secondary antibody to test the expression of the protein in each sample. So over, you can see the picture is, here's the total protein constant expression. So this is the total sheet, and CT and BD, you can see that there's a slight decreasement in the protein concentration in BD versus the protein concentration in the CT sample. There are some further results. So BAX is a protein that promotes cell death, and BCL is an anti-apoptotic function, so you can see that they both have decreased levels in the BD. Furthermore, there this is the expression of eNOS, and eNOS has a protective function in the cardiovascular system, which can be attributed to NO, and this regularization of the vascular tone is a common, best known role of NO. And after BD, the SNO levels are lower, and so there's no need to produce NO, so that's why eNOS is increased in BD as opposed to CT. So that's why that graph is different than the others before. So after analyzing all the graphs, the hypothesis was proven right. Big picture wise, brain death is negatively impacted, biological activities are negatively impacted by brain death, and that was proven from the decreased SNO levels, this big change in the two methods to collect tissue oxygenation, and the decreased protein expression of the Western blot. So here's some pictures from my summer. It's really cool working in the lab. I really loved every day waking up, feeling like it was really cool. You know, you had your own badge. You got to go into the lab. It was really a great experience. We got to wear white coats. I think that was like one of our favorite. We got the white coats.
The gloves.
And I just want to take some time to thank my mentor, Dr. Lin Zhu. You made this summer really fun and really interesting, and I'm really appreciative of all you've done.
Hi, I'm Gabby. I'm a junior, and I did organ donation with brain dead patients this summer, and my mentor was Mohammed. So before I got to start my work, I had to learn about the different types of donors first. So there's 3 different types: living, cardiac dead, and brain dead. So a living donor is typically in good physical and mental health. They usually have a BMI less than 35, and they must be free from uncontrolled high blood pressure, diabetes, and cancer. And then cardiac death, just the basic definition, is death caused by sudden heart failure. And then brain death, which is what I mainly worked with, is the irreversible loss of all functions of the brain, including the brainstem. And so I also had to learn about the monitors for brain death, and there are 2 types that I worked with, which are the intraspectra and the T-stat. So the intraspectra measures tissue oxygenation, and it indicates hypoperfusion as well as basic deficit or lactate in trauma patients. And the monitor is placed in the thenar muscle, which is located on the ball of your thumb, which is like right here. And then the T-stat monitor measures oxygen saturation by using blood from the capillaries, which is also called microcirculation, and that is placed orally, so it's like on the inside of your mouth. So my specific case that I worked with was a 51-year-old male, and he was at Metro Health, and so I got to learn about the patient, and so he committed suicide. He was a heavy smoker. He also did drugs like heroin, crack, cocaine, and marijuana, and his drug addictions were the main cause of his depression. And he also had previous suicide attempts. And so, like I said, he was at Metro Health, and then the other hospitals that we got donors from are University and Cleveland Clinic. And so my experience, that donor, I had muscle samples from him, and I prepared them, which just means I lysated the muscle, which means you break it down, and then you mix it with different kinds of antibodies, and then you place it into a 96 well plate. And then, so that was just a way to find protein analysis. And then you also, like Brianna's presentation also had this, it was like a gel plate thing. And so basically you would transfer the protein onto a gel, and then you would transfer the gel onto a membrane, and then you'd scan it, and this is the results. So the top one was the target protein that I was looking for, and then the bottom one is GAPDH, which is just like your baseline. And so as you can see, there was a good amount of protein in the samples. And so, yeah, also we got blood samples from the donor just to see the different hemoglobin levels. So every day I came in, I did a Western on them, and it's a 3-day process, so I just did that every single day, and I got really fast at it, so thank you, Mohammed. And then I got so good that I got to teach my other colleagues, which is a blast. And so yeah, my first week and a half, I had no idea what I was doing. I just kind of sat there and listened to Mohammed. He taught me the basics, and then I just got really fast at it, and I knew what I was supposed to be doing. And so the cool thing about my project was I got to experience both basic and clinical, which just means that I got to go into an ICU and like see the patient. I got to see the monitors, and I got to see like the blood samples that we took, and then I also got to do lab work on the samples. So now I'm kind of leaning towards a STEM career, which is really cool. And then these are just some pictures. So that was for our pig surgery, the ones in scrubs, which we thought were so cool. This was the first day that we met our mentors, which we just were so excited. We were just so dorky. And then this was the first day in the lab, and we got to see the pig that we were doing surgery on, and we had to wear like the hairnets and the little covered thing. And so, again, special thanks to Mohammed. This was our last day when we went to Chipotle and Mitchell's, and it was a really fun time just to bond with our mentors and get to know them a little better outside of the lab. And so thank you for all the food that you bought me.
Hi, I'm Elena Sturgill. I'm a junior here at Beaumont, and this summer I worked with my colleague / mentor, Edwin, in the back, and we worked with snow hemoglobin synthesis. So, snow hemoglobin is an important protein in human bodies. It is responsible for transporting oxygen in blood. Hemoglobin is one of the many proteins that can be nitrosylated, which is why the lab as a whole uses it. And the nitrosylation process is valuable because it acts as a general mechanism for control of protein function and vasoregulation. So in order to conduct this experiment, there has to be focus on a specific pathology. So type 2 diabetes is a long-term disorder that is a result of poor eating and exercise habits. And it occurs when the body has a problem managing its blood sugar levels and cannot make insulin effectively. There's currently no cure, and people have to manage their diabetes to keep blood sugar levels within a range. It can go unnoticed for years, and it can be prevented with a healthy lifestyle such as eating well and exercising regularly. So 29.1 million people are affected by type 2 diabetes, and so that means 1 in every 10 adults 20 or older has diabetes, and then 1.4 million new cases every year in the United States alone. HbA1c is a type of hemoglobin that is bound to glucose. Glycated hemoglobin is a form of hemoglobin that is measured primarily to identify the 3-month average plasma glucose concentration. And then the duration of this test is limited because the lifespan of a red blood cell is 4 months. So my preliminary data is HbA1c is inversely correlated to snow hemoglobin. So that means as the levels of HbA1c increase, the levels of snow hemoglobin decrease, and vice versa, which can be seen in here. So the main question, does HbA1c get nitrosylated? As mentioned before, this summer I only used snow hemoglobin to see if this experiment is repeatable and could be used on HbA1c. So if HbA1c does become nitrosylated, it explains the inverse trend, but if it does not become nitrosylated, then further research needs to be conducted. And we attempted to nitrosylate in vitro, which means in the cell. So my protocol, we started off with equilibrating, or washing out the columns with each specific buffer. So this on the right is washed with borate buffer, and then the one on the left was washed with PBS EDTA. So you would pour and collect the hemoglobin, and then measure the hemoglobin concentration through that. And then we use this machine called Perry, which measures the hemoglobin concentration using light, and it identifies the substance as hemoglobin, oxyhemoglobin, carboxyhemoglobin, and methemoglobin. So you would put what came out of the column into a little thing, like cube thing. Then you put it in here, and then what would show up on the screen would identify what the concentration of that small substance is. And then, so the last machine that was used in the experiment was called the EPOC. It analyzes the 96 well plate using light to find concentrations. So, in non-diabetic patients, there is no correlation between HbA1c and snow hemoglobin. And these are patients in cardiac pulmonary bypass, and if the patient is below that range, there is no correlation between HbA1c and snow hemoglobin. HbA1c correlates negatively with snow hemoglobin in diabetic patients. We can see an inverse correlation between snow hemoglobin and HbA1c, which led to a further question, could we say anything about a patient that is considered diabetic? So next summer, Edwin. Okay, this graph shows the straight line is the standards curve, while the dots are the single points that we found, the little samples, those were the amounts, the concentration amounts. So this just compares the standards and then what we found. And then, so theoretically, there should be 2 snow per tetrameter of hemoglobin, but experimentally, after many repeats of the experiment, the closest number to 2 that we could get was 1.6 and 1.7, which is still repeatable and good information. So I decided to join this internship because the only thing I would be doing this summer is working a minimum wage job, sitting at home on my couch, watching Netflix. So this I thought would help boost my resume, allow me to work in the lab, come closer to my friends, and it's useful in networking, and it helped me realize that the STEM career is something I am very interested in pursuing. And I noticed that in the beginning of the internship, I was really scared to use a pipette, and I was really nervous, but now I asked my mom for my birthday to get me pipettes. And then again, the picture of us eating. And then thank you to Edwin and everyone else at the HDI Institute.
Hi, I'm Izzy Valcito, and I'm a junior here at Beaumont. And my project was, or our project was, assessing the viability of stem cells from brain dead patients. And my mentor / colleague is Dr. Arshana Qureshi, who cannot make it. She stings a little bit.
Next time.
All jokes. So this summer I had the amazing opportunity to work alongside my friends at the Harrington Discovery Institute. And as I said before, my mentor was Arshana Qureshi, and she studies hematopoietic stem cells. So there's a severe shortage of organ donors in the U.S., specifically bone marrow donors, which is a pretty tricky transplant process. And so this led to the question, could bone marrow be obtained from subjects after brain death and still be viable? So the main objective of the project was to evaluate the functionality and viability of hematopoietic stem cells obtained from the bone marrow of a brain dead subject by the use of glycol centrifugation. So here's some background on hematopoietic stem cells. So basically it differentiates into the different components of your blood, like platelets, white blood cells, red blood cells, plasma. Here's like a little chart for differentiation. Like the nickname is blood stem cells, which is a lot easier to say than hematopoietic. So this is the technique that we used. So the goal basically was to separate, there we go, the white blood cells from the sample. And the way we did this was glycol centrifugation. So we took a bone marrow sample from a brain dead donor, and you place it in a centrifuge tube, which is like, if you know what a test tube looks like, it's like that with like a pointed bottom. And so in the centrifuge tube, we put in PBS, which is a buffer, and a glycol solution, which is actually pretty poisonous, so I had to be pretty careful about that. I'm pretty clumsy, so that was a process. And so after you set up the solution, you centrifuge it for 40 minutes. And a centrifuge, it looks pretty unassuming, but it's like a giant box, and you open it and place a tube inside, and it whirs it around to layer the solution. So we centrifuge that for 40 minutes to separate it into the plasma, red blood cells, white blood cells, and the extraneous buffer, which is also called the supernatant. And so after the first centrifuge, you remove the supernatant with a pipette, which is pretty difficult because it's like this top layer right here, and it's right above what you're trying to extract, and it's like super, super thin. And so when I first started out, I had to use like old samples because I messed up at least 20 times. So I couldn't like start with the important stuff yet, at least 20. It's probably more. Yeah. Okay.
Okay.
Yes. So after you have your new layered sample, you add more buffer into it, and then you mix it up with your pipette, and then continue the centrifugation process. And about after 3 times of centrifuging the sample, you get a small layer of white blood cells, a small layer of red blood cells, and a little bit of plasma, which it's like probably 3 centimeters in height, which is pretty small. So the white blood cells is what you're trying to isolate, and they contain a marker for CD34 stem cells. And after you take out the white blood cells, you analyze the sample using a flow cytometer, which it's basically a laser that you stick your sample into, and it'll count the markers on the white blood cells. And if there's enough markers, then you can see if the sample has any traces of viability. So here's a real life picture. So it's a plasma. You can see it from there. It's like the little layer of white blood cells, and there's some more extraneous solution right there. And so after we got the samples, I had to plate them on little petri dishes. And so after that, you have to incubate them to let them grow. And at different stages of incubation, I had to count them. And it was like on a grid system. And there are 2 different types that I counted: GMUs, BFUs. You can see the difference between there. So like the grids was about 200 on each plate. And I had to go square by square to count the different kinds, and took a solid hour and a half each time. So my experience. Going into the internship, I had 0 lab experience at all, and I had no idea what I was doing. And so I wanted to see if I would enjoy it. And coming out of it, I got actual lab experience. I learned different techniques, and I got to like contribute to an actual project. And I think Archana has 2 grants now, right?
Just
No. Okay, the project, I guess I should say, has 2 grants now. So
project is fun.
yeah. So it feels good to like contribute to an actual scientific project. And I got more friends. And now I have a new interest in the medical field. So thank you to Archana, who's not here, and everyone else at HGI for making it a nice, fun summer.